So again, like it's really small. It's just like I think more yeah, I know. Well, there was like I think so too. This is the molecule I did my undergraduate. But yeah, so just
Okay, let's get started. Only have a little bit of material left. So now that we've also become expert in female reproductive physiology, today's lecture will kind of focus on getting male and female stuff together and see what happens. Okay. So my plan is to cover fertilization today, pregnancy, parturition, which means childbirth, and lactation. Okay. And then tomorrow we'll cover something called IVF. Anyone want, what's IVF? IVF? In vitro fertilization. So basically tomorrow will be the outside of the body experience of what we're talking about today. Okay. Okay, so yesterday's stuff, female reproductive physiology, the monthly cycle. Any questions about this material? It's pretty straightforward. What's easier, male reproductive physiology or female reproductive physiology? Male by far, right? It's nice and easy, it makes a lot of sense. Female reproductive physiology involves hormones going up and going down, going all over the place. Okay. So when you're kind of preparing for this, for the exam, you know, there are a couple of slides that have some overlap and stuff like that. So what I would do is try to picture what the follicles look like. And that slide that has the ovarian phases and the uterine phases right there for a whole month, and kind of talk to yourself, try to figure out, okay, what's happening early on? What needs to go where? Okay, what's happening in the ovary? And whatever happens in the ovary is going to impact the brain, and it's going to impact the uterus. Okay, so once you kind of keep having that stuff in your head or kind of visualize it, some of it should hopefully make some sense. Okay, let's see. What happens about 7 days into the reproductive cycle? Ovulation? Who says yes? At least somebody said yes because they said ovulation. So what else? Anyone else? Don't leave them hanging. Come on, help them out.
One follicle become dominant.
I like that one, right? So if you want to split it in halfway, ovulation is the midpoint. Okay, so about midway through the follicular phase, right, which is half of the reproductive cycle, we exactly have that. We're separating out the one that is destined to ovulate from the other ones that for some reason are no good and they end up dying off. Okay, so ovulation at about 14 days. So if you kind of think about it, we had all these graphs, right? 28 days, that's the female reproductive cycle. So let's start January 1. On January 14, you ovulate. Okay? And this is what ovulates. Okay? We kind of talked about it yesterday. What can you tell me about that egg that ovulates? Can also be a very fair answer would be, no idea, I don't need to know it until next week. Anything. If I were to give you a hint, something related to meiosis.
Has 23 chromosomes?
23 chromosomes, exactly. And what has completed and what just started? Meiosis 1 completed, and meiosis 2 started, and meiosis 2 arrests. Okay? And exactly right, 23 chromosomes. And in addition to that, what else could I find? That it's kind of stuck between the egg membrane and the egg shell. The polar body, the first polar body, okay? So what can I find inside that secondary oocyte, because it's in meiosis II, so secondary oocyte, and the polar body? Something like this, or something like this? This one? Ooh, we have some disagreement, I like it. Okay, so this is in a secondary oocyte, right? Okay, the suspense is killing everybody. This is in a secondary oocyte and also in a first polar body. What can I find? What was the other stimulus that causes completion of meiosis II? Because we arrested meiosis II now, the egg is floating around in the fallopian tube. We need another signal to complete meiosis II. Fertilization. And the result of fertilization is completion of meiosis II and another polar body. What can I find at that point in that secondary polar body or the second polar body? Which one can I find? This one or this one? This one, right? Make sense? Follow the chromosomes, follow the chromatids. It's like a game, okay? Okay, so let's say if fertilization were to happen, okay, the egg is ovulated, this thing that is floating around in the fallopian tube or the oviduct is viable for 48 hours, 2 days. That's it. After 2 days, if it's not fertilized, it will die off, okay? Sperm, if it is present in the female reproductive tract, is viable for about 72 hours, 3 days. So let's go back to our calendar. On January 14, you ovulated. 14, 15, and 16, that is a viable egg. So just make sure sperm is around covering those days. Okay, that leaves me lots of days where fertilization cannot happen, right? How well does that work as a contraceptive? Very, very bad. It's not a good idea. Why is that? Is a reproductive cycle exactly 28 days? No, it's not. Okay? It can be 28, it can be 26, it can be 32, and then it's 31, and then it's 28 again. Okay, it's variable. The only reason we put 28 days is because we need to have some framework to kind of go through all these different things that are happening. Okay, so generally speaking, that is not a very reliable way of preventing pregnancy, trying to avoid about 4 days where you figure ovulation is happening. Okay, anyway, let's talk about fertilization. Okay. This thing is what ovulates. Remember when we talked about, we have that mature dominant follicle. It was a gigantic structure. It's actually that egg and the surrounding cumulus cells that leave, and the rest that stays behind from that follicle, that actually reorganizes into the corpus luteum, right? So these cumulus cells are actually kind of held in this gooey matrix, okay? They're not like a brick wall surrounding the egg. It's just kind of like a gooey gel that has some of these cumulus cells, and that's kind of floating around. And then we have sperm that actually has to get through that and fertilize that egg. Okay, this is a picture of a female reproductive tract again. Okay, here we have the vaginal canal, the cervix, the uterus, fallopian tubes, okay? And for some reason from this paper, they didn't care for the ovaries, so the ovaries are not listed on there. Now, in all fairness, if we're going to talk about fertilization, we don't care about ovaries anymore because the egg is out anyway, okay? So how many sperm need to fertilize an egg? 1. How many sperm actually enter the vaginal canal in an average ejaculate? Millions. Okay, so we have a little bit of stuff going on here, okay? So the first thing that we're going to talk about are barriers to fertilization. Okay, so there are clearly physiological and anatomical barriers to fertilization. And then we're going to talk about something called block to polyspermy. A block to polyspermy means there's things that are going to happen that prevent multiple sperms from fertilizing that egg. And obviously, some of it what does help is the barriers to fertilization. If you look at this figure right here, the first barrier to fertilization is the cervix. Okay? The cervix is an anatomical barrier to fertilization. If there are, let's say, 300 million sperm in an ejaculate residing in the vaginal canal, most of them will never pass through the cervix. Okay? There's a lot of side grooves and in the cervix, and sperm just kind of get lost. Especially sperm that, for example, are a little bit abnormal, either maybe a kinked tail or there's something, other structural abnormality. There's no way that ever gets through the cervix. Okay? So the first barrier to fertilization is the cervix. The second barrier to fertilization is a little further higher up. Okay, so we have a couple of thousands that may make it through the cervix that are in the uterus right now. They're moving upward. What is responsible for that movement? Smooth muscle contractions. Sperm are not swimming. Okay, so it's actually smooth muscle contractions here that will help move the sperm further up. The next barrier is right here. This part here or this part right there. And it's called the uterine tubal junction. Okay, or UTJ, uterine tubal junction. And really, it's kind of what the word says. It's that junction, that area where the fallopian tube is connected to the uterus. Okay? Again, that's an anatomical barrier. Even with inside the fallopian tube, so the ones that actually make it through that, this is a cross-section of a fallopian tube. That tiny little pinhole in the middle, that's the lumen. All that other dark stuff, those are all glands and places for sperm to get lost or stuck or whatever. Okay? So if fertilization happened on this end, only those few sperm that actually made it there, maybe about 100, have a chance of fertilizing that egg. So we started with 300 million. Now we're talking about maybe 100. So a lot of sperm never make it to that point. Okay? And then obviously sperm, when they're here and they have to make a decision, I either go this way, which is the good way, or that way, which is the wrong way. You know, they don't know where to go. So maybe it's like stupid sperm go this way and the smart sperm go that way. Who knows? Okay? So those are kind of the barriers to fertilization. Any questions about this stuff? Yes?
Can you point out where the UTJ is?
It's the uterine tubal junction, so it's the junction between the fallopian tube and the uterus. So here's one, and there's one. Okay? Any other questions? Make sense? Okay, perfect. Okay, the one thing that's going to happen, there was a structure in the male reproductive tract where sperm matured. Do we remember what that was called? There's not that many structures, so. Starts with an E. Epididymis, perfect. Epididymis, there was some sperm maturation, right? You could actually take a sperm from that area once it's reside there for a while, like from the tail, and if you were to put it in a dish, that thing actually could start swimming. Okay? Now there's another place where sperm is going to continue maturing. It's like a second maturation step. That's in the female reproductive tract. Okay? That's actually right here. Okay? And that maturation step is called capacitation. So I put it in bold and in red. I expect you to know that term. Capacitation is another maturation step, or the final maturation step of sperm. And the things that are going to happen here, there's two things. Okay? So capacitation involves two things. One, we see an increase in membrane fluidity. And we're talking about the sperm membrane. So when sperm resides there for a while, we're going to make the membrane of the sperm head more fluid. Remember like a decade ago when Dr. Warren talked about cell membranes? I know he did. So if you don't remember, I'm sorry. Next week, that comprehensive final, it sucks. Okay. Do you remember anything that you could put in a cell membrane that makes it more rigid? Cholesterol. Okay, so in fact what happens here is basically you can strip some cholesterol out of the sperm membrane, and make that membrane a little bit more fluid. And the reason why you want this is because you're going to be fusing two cells together. If you make the membrane a little bit more fluid, that fusion event is more likely to occur. If you make the membranes really rigid, okay, it's harder for the cells to fuse. Okay, so that's the first part of capacitation. The second part is hyperactive motility. So now at this point, the second part of capacitation, right in the fallopian tube, the sperm finally starts beating its tail like crazy. Okay, it senses that the ovulated egg is nearby, and now it's going to start using its mitochondria and generate a lot of energy to beat its tail like crazy. And what has to happen then, it needs to go through that the cumulus cells are not like that anymore. They're not like a brick wall. Okay, they're kind of more spread out in this gooey substance. Okay, so hyperactive motility. Okay, so we keep going. What's the first thing, once sperm get through all the gooey stuff and the cells, what's the first thing that the sperm hits after all the cells? The zona pellucida, okay, the zona pellucida, the eggshell. Okay, and think of the eggshell as this gigantic matrix, but it has some receptors for sperm. Okay, so sperm can actually bind to that eggshell. And when the sperm binds that eggshell, we have something called an acrosome reaction. The acrosome, remember, that was in the sperm head, and I told you to think of it as a garbage bag with enzymes, digestive enzymes, okay? So once sperm actually hit that eggshell, we get what is called the acrosome reaction, and all that means is the acrosome reaction is that all these digestive enzymes are being released, okay? So picture this little torpedo now with a long tail, beating its tail like crazy. You get all these enzymes being released to digest a hole and drill a hole through the eggshell. Okay? So that's why we need the acrosome reaction, to make that hole. And then it takes about 5 to 10 minutes to actually get through the whole eggshell, and then you're in that space between the eggshell and the egg membrane. Okay? Make sense? Any questions about any of the stuff I just mentioned? So we know what capacitation involves and where that happens. We know what acrosome reaction means. Okay, so then we finally get this. Okay, fusion of sperm and egg. So here, so they didn't draw the zona pellucida around here. Okay, but here you have that sperm head lining up with the oocyte membrane. And at this point again, we have maybe 100 sperm that are really racing right now because we can only have one fertilizing the egg. Something called polyspermy, multiple sperm fertilizing an egg, does not work in humans and in most mammals. Okay? It causes chromosomal chaos. It cannot develop into an embryo then. Okay? As soon as that happens, we get a sperm nucleus entering the egg, and this causes completion of meiosis II. At that point, some of these sister chromatids are getting pushed out into a second polar body. Okay? Now, because each of these, you have the male sperm nucleus and the egg nucleus, they're often referred to as a pronucleus. They're not really quite a full nucleus yet. Each of these have 23. They still need to combine and form a 46 chromosome containing nucleus. Okay? So each of these are pronuclei, a male and a female one. Okay? Once this happens, we are called a zygote. We are a single cell. Okay, still with a surrounded eggshell, and this will give rise to everything. The whole embryo, the placenta, all that stuff. One cell, one nucleus, 46 chromosomes. Okay. Make sense? Cool. Okay, like I said, what if 2 or 3 sperm are really close together? How do we prevent some of the other ones from fertilizing that egg? and that is called the blocked polyspermy. Okay, so the blocked polyspermy is kind of highlighted right here. Okay, so we have a sperm, many sperm bind receptors on the zona pellucida. You get the acrosome reactions swimming, they move through the zona pellucida. One sperm at some point timing, it's the fastest one, that one will actually touch the egg membrane, start fusing, and that initiates the blocked polyspermy. And the blocked polyspermy is two things. Luckily, it's only two things. Okay. The first thing is that we have a change in membrane oocyte potential, or membrane potential of the oocyte. Okay. Basically, we're changing the oocyte, the egg membrane, such a way that if there's a sperm right behind, it cannot fuse anymore. That fusion event is not happening. Okay. So that's good, right? If these two are close together, that second one is just a couple of seconds too late. It cannot fuse anymore. Okay. The second thing is something called the cortical reaction. The cortical reaction is the egg's counterpart to the acrosome reaction. Okay. So what we have is we in fact have an egg here. Here's the zona pellucida. And near the periphery of that egg, we have a whole bunch of vesicles. And those are called cortical granules. Those vesicles are also little garbage bags with enzymes. Okay. So the egg now releases a bunch of enzymes. And what these enzymes are doing is they harden the eggshell. They make that eggshell super hard. So if there's still a sperm that is obviously way too slow, if it starts touching the zona pellucida, nothing's happening. Those digestive enzymes are not degrading anything anymore, and there's no way for that sperm to start entering through the zona pellucida. If there is a slow sperm that is all halfway through the zona pellucida, that zona pellucida, the eggshell gets so hard, it just gets stuck. It stays there. Okay. So does that make sense, how you can have changing the membrane potential and something called the cortical granule reaction or the cortical reaction, that it prevents multiple sperm from fertilizing the egg? Make sense? If it doesn't make any sense, speak now or forever hold your peace. Okay. Second, pronuclei fuse, second polar body, blah, 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 embryogenesis. That's it. Again, if none of this stuff makes any sense whatsoever, you have my email address. Okay, now we're going to do a little bit of developmental biology. Okay. So I told you fertilization happens right here in the fallopian tube. Okay. So in fact what happens is the ends of the fallopian tube, they have these finger-like extensions. Okay. And kind of picture these fingers waving over the surface of the ovary. So anytime there's an ovulation, it actually captures that egg and puts it into the fallopian tube. Okay. Then we have fertilization happening and we are a zygote. Normally, that zygote, okay, turns into a 2-cell embryo. 2-cell, 4-cell, then an 8-cell, 16, 32, 64, 128. You know where this is going. And as all this stuff is happening, that embryo is slowly moving through the fallopian tube, through the uterine tubule junction, and into the uterus. Okay? By the time it enters the uterus, it is actually called a blastocyst. Okay? So a blastocyst is this embryo. Okay? That is actually the one that is going to attach to the endometrial lining and invade that tissue. Okay? So what would happen if that fertilized egg actually gets stuck here in the fallopian tube and it doesn't migrate down?
Would that be an ectopic pregnancy?
Ectopic pregnancy. Okay, so an ectopic pregnancy. Okay, I think it's about 1% to 2% of the women in the U.S. would experience an ectopic pregnancy, and basically in most cases, the predominant cases, that fertilized egg right here just gets stuck. And as it continues to develop, it's going to, instead of invading here, it's going to try to attach to the lining in the fallopian tube and invade there. Now, I hope you remember from yesterday, the hormones from the corpus luteum spend a lot of time making the endometrial lining really thick, and you can attach and invade there. Not so much here in the fallopian tube, that epithelium. Okay, you can't really invade any of that tissue. So usually those are extremely painful, causes a lot of bleeding, and if it's catched pretty early on with hormone treatment, you can actually get rid of it. Otherwise, you have to surgically remove that. Okay, it's dangerous. Now, let's say you have that fertilized egg, and instead of it getting stuck here, it actually falls out this way. So, bloop, out of the female reproductive tract. What would happen?
Pregnancy in your kidneys.
Pregnancy on your kidneys, maybe not in, but on your kidneys, on your small intestine. Think that's possible? No? That sounds like science fiction, right? Okay, so in very, 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 very rare cases, okay, it does happen, okay? And you can actually end up developing something called lithopedians, or stone babies, okay? Most of these cases, you know, these are really small. It never develops into anything. And these are just little calcium stones that disappear. In rare instances, we'll have something like this, okay, where you get partial development, and because it dies, you get a lot of calcium deposition, and these become completely calcified, okay. So here's one example. Here's another example. There have been only less than a handful of publications in very obscure medical journals that have actually reported near-term carrying of an abdominal pregnancy, and actually through a C-section, a healthy baby, okay? But again, most of the time that never happens. It is kind of fascinating though, right? How that embryo, in very limited certain cases, could actually find a niche and actually set up something that it will get enough nutrients and actually develop into something like that, okay? If that were really possible, okay, but hypothetically here now, science fiction, what would that mean? We could make pregnancy so much more interesting, couldn't we? Because at that point, if this was true and you could really do this quite normally, I think it's a time for men to step up, right? We might as well take a fertilized egg and put it in the abdominal cavity of a male and have it carry everything to term. Why not? Okay. So I can tell you already that men are not very excited about this whatsoever, okay? Men are not built like women. Men handle pain very poorly, okay?
I'm a little baby for science.
Yeah, for science, exactly. Okay, let's look at this. All I'm doing here right now, again, is adding just a little bit of terminology, okay? So again, this is a fertilized egg. This is a zygote. How do you know that this figure here is a zygote? So panel B. So yeah, sorry, say that again? You have what? 2 pronuclei, perfect. That's one, and what's the other hint? It's a little bit more difficult to see. 2 polar bodies, okay? So when you look under a microscope, okay, if you see something like this, okay, you know that that's not a fertilized egg. You don't see 2 pronuclei, and you don't have 2 polar bodies. This is a zygote, a 2-cell embryo, 4 more, even more, lots of them, and then finally a blastocyst. So the ones where you really have lots of cells all compacted, that's referred to as a morula, okay? And that morula embryo then develops into a blastocyst. So the blastocyst, think of it as something like this. You have a single layer on the outside that is called the trophectoderm. Trophectoderm. Okay. The trophectoderm is comprised of a single cell layer, and these cells are often called trophoblast. Okay. That part gives rise to the placenta. Okay. And then inside you have a clump of cells that is referred to as the inner cell mass. Those are actually the embryonic stem cells. So for those of you that heard of embryonic stem cells before, maybe in the media or in science press or whatever, that's where that comes from. The embryonic stem cells gives rise to everything that currently, as we're in this room, makes up your body. Everything. Okay? The only thing it doesn't give rise to is the placenta. The placenta comes from the trophectoderm. Okay? And then this whole cavity here, that's all blastocele. That's just a fluid-filled cavity. So this is the structure that is now going to implant. Okay?
So, quick question. So for the morula, is there like a specific number of cell divisions that have to be reported?
That's a great question. And so we always cheat. And again, me lying. Like when I say it's a 1 cell, 2, 4, it doesn't quite go that way. You can actually have 5 cells if you time it just right. Okay. So how many cells are morula? I think usually they stop counting after 128, but at that point really, it's just when it becomes very compacted. Yeah. So again, remember from the first day of class, we kind of had some fun how crazy humans were thinking about depending on where the embryo attaches, then it turns into a male or a female. So this actually kind of shows you the normal implantation sites. It really doesn't matter, okay? Left, right, top, okay? And it has nothing to do with sex determination, okay? And again, this is what it looks like. Here's the inner cell mass. Those are the trophoblast cells. And then right here on the bottom, it's going to attach to the endometrial lining and invade. These cells will proliferate, invade. They change the vasculature. In fact, at one point in our life, when we were a blastocyst, you can almost think of us at one point in our lives, we were a cancer to mom, okay? We attached to her endometrium, we invaded all that tissue, we changed the vasculature. I might as well be talking about an aggressive tumor, okay? The only difference is, in this case, mom's body is very well capable of dealing with this and making sure this doesn't spiral out of control, okay? Okay. Any questions about this stuff? Okay, the one thing I want to point out now, the blastocyst, it takes about 5 to 7 days to become a blastocyst after fertilization. Okay, let's see what we remember from yesterday. Where are we in the monthly cycle at this point? Because this becomes important. Let's start with this. Remember how we had like 2 ovarian phases? A follicular phase and a luteal phase? Which of those are we? Which one? Your odds are pretty good. It's like 50%. I swear to you, you don't get better odds on the final exam. Luteal, the luteal phase, okay, right? Because it's after ovulation. This is 7 days after ovulation. Okay. Now, recognizing this, we have what in the ovary? The luteal phase. We have a corpus luteum. We have estrogen, progesterone, doing all that stuff. That really important stuff, right? What's going to happen in a couple of days as we're this?
No period.
Sorry?
No period.
Yeah, but what normally would happen if we don't do something? Yeah, the corpus luteum is going to die, right? Because it only survives for 11 days. So what has to happen? If this is happening, we need to tell mom she's pregnant, right? So in fact, that embryo has to tell mom that it's there. So it has to send out a signal to show that it's there. And we need to maintain that corpus luteum. The corpus luteum has to survive. It has to keep secreting progesterone and estrogen. Because what happens if it does not? What happens if I lose progesterone from the corpus luteum? What happens to this thing? Yeah, miscarriage, sloughs off, you lose it. Okay? Make sense? So that signal, that event is called maternal recognition of pregnancy. Okay? So maternal recognition of pregnancy. And in fact, what happens in humans, since this is human physiology, so we're going to ignore all the other mammals, some do it a little different. This trophectoderm, or the trophoblast cell, in fact will secrete a hormone called HCG, human chorionic gonadotropin. Human chorionic gonadotropin. So HCG from the trophoblast cells gets secreted, goes into mom's bloodstream, goes to the ovary, and ensures that the corpus luteum survives and doesn't start dying. And that's how we maintain high levels of progesterone. Okay? Make sense? So it's kind of shown here too. With the HCG, we maintain high levels of progesterone and estrogen. Okay. Do you guys know how a pregnancy test works? What do you do with a pregnancy test? You go to Walgreens, you buy it, or Walmart, whatever. You take it home, and you do what? You pee on it, and then you see a smiley face or a frowny face or whatever the hell happens. Okay? So what effect are you looking for? What is the test looking for? Progesterone? But if it looks for progesterone, what is the test telling you? It could tell you that you're in the luteal phase, and you're not pregnant. But that's not going to be helpful. You do not want to find out that this is a false positive. HCG, you're measuring, you're looking for HCG. Okay, so basically the pregnancy test actually look for the presence of HCG in urine. Well, you guys are all experts in renal physiology, and you know how HCG ends up in urine, right? Just filtering blood, that's it. Okay. So HCG is very similar to another hormone that we've talked about, called LH. And in fact, LH and HCG are identical, except where they're being made is different. HCG is made by the embryo, LH is made in the anterior pituitary gland. Okay? The other difference is that HCG is glycosylated. And if you remember from maybe the first week or 2 weeks from class, sometimes we add things to proteins called post-translational modifications. And one of these things was glycosylation. You add sugar groups to it, that makes it more stable. So it's around for a longer period of time, okay? And that's the difference between HCG and LH. HCG, coming from the embryo, is glycosylated. So it's more stable, okay? There's a longer half-life. Okay, how long is HCG around for? Not horrendously long, okay? So in fact, what happens is for the first couple of months, this HCG really needs to be there to maintain the corpus luteum. So we keep cranking out more and more estrogen and progesterone levels are always higher than estrogen, okay? But then after a while, we get rid of the HCG because in fact, what's going to happen, we don't even need the corpus luteum anymore because now we've developed a placenta. And the placenta will start taking care of all the progesterone and estrogen that needs to be made. But we need some time for the placenta to develop, okay? So early on in pregnancy, it's the corpus luteum that secretes estrogen and progesterone thanks to HCG. And then once we have a fully functioning placenta, the placenta will make all this estrogen and progesterone. And it keeps making more and more and more and more, okay? The reason why we want progesterone levels always higher than estrogen levels, well, progesterone inhibits myometrial contractions. Estrogens promote myometrial contractions. So we always want progesterone levels higher than estrogen levels. Make sense? But you need both, okay? Any questions about this stuff? Okay. So just really briefly about the placenta, my favorite organ ever. Okay. This light pink on your left is maternal part, so that's the endometrium. This dark, it's kind of like purple, that is blood. And this light pink is the fetal portion of the placenta. Okay. So what started out as a single cell layer right here turns into what is now light pink right there. And it's kind of like a nice disc shape. It doesn't cover the whole uterus, like what you would, for example, see in horses, but it forms kind of like a frisbee, kind of a disc at one part of the, one part in the uterus, okay? So two main functions of the placenta, you need to provide oxygen and nutrients. You also have to get rid of waste and carbon dioxide. And the second part, endocrine function. I just mentioned it's a huge source of progesterone and estrogen. And you need that to maintain the pregnancy, okay? We cannot have any mixing of blood because the fetus is genetically different from mom. It will cause an immune rejection, okay? So basically, the way I want you to picture that fetal portion of the placenta is you have these tree-like structures, okay, called chorionic villi, highlighted right here. Okay, and basically that's maybe a single cell layer thick that is bathing in mom's blood. So that exchange from mom's blood here into fetal blood, okay, happens very efficiently. And then we have all that blood going through the chorionic villi. It all gets collected into the umbilical cord. Now, in the umbilical cord, we have an umbilical vein and an umbilical artery. And this is where things get confusing. Because in fact, it's the umbilical vein that carries oxygenated blood and nutrients to the fetus. Okay? And it's actually the umbilical artery that takes oxygen poor blood and waste from the fetus back to mom. Okay? So the umbilical vein has the oxygenated blood carrying it to the fetus. Okay, after 9 months, trust me, most moms will be very excited to have the baby come out. Parturition, okay, or labor, okay, childbirth. So what happens here is we're going to introduce more hormones. I told you, reproductive physiology is all hormones, okay? So at some point during pregnancy, the fetus grows. So what do you think is going to happen? Well, the fetus is going to keep pushing on the cervix, okay? So near the end of pregnancy, the fetus drops lower into the uterus, pushes on the cervix, and we cause something called cervical stretch. Okay, so basically we start stretching out the cervix. This is causing feedback to the brain, positive feedback to the brain. And in fact, it causes the posterior pituitary gland to secrete oxytocin. So there are only 2 hormones coming from the posterior pituitary gland. One was vasopressin, that has nothing to do with this, it's kidneys, and the other one is oxytocin. Oxytocin is important, okay? Oxytocin, in fact, goes from the posterior pituitary back to the uterine wall. And not only does oxytocin act on the uterine wall, but I want you to put here next to it is estrogen. So the estrogen that the placenta secretes also acts on the uterine wall. And it's both estrogen and oxytocin that causes the uterine wall to secrete prostaglandins. Okay? And prostaglandins stimulate contractions, uterine contractions. Okay? Now, what happens if you contract the uterus? Well, you push the fetus more on the cervix. You get more cervical stretch. You get more oxytocin from the posterior pituitary gland. You get more prostaglandins. You get more uterine contractions. I think you see how these positive feed-forward loops keep going and going and going. Okay? Make sense? That's parturition. That is getting the baby out. Now, if you look at this hormone profile again, okay, throughout the whole pregnancy, we see that progesterone levels are higher than estrogen. I told you that progesterone inhibits myometrial contractions. How is this possible then? All right, so how come we still have contractions with progesterone levels always higher than estrogen levels? And this is something called functional progesterone withdrawal. So even though progesterone levels are always higher, think of it as after 9 months, the progesterone receptor gets so tired from being bombarded by this hormone, it doesn't listen anymore. Okay, so it stops listening as well as it did earlier on in pregnancy. So that is functional progesterone withdrawal. Okay? And then what happens all of a sudden? How come progesterone levels and estrogen levels go from super high to 0?
You're not needed anymore.
You're not needed anymore, but practically, what is causing that drop? Sorry?
Loss of placenta.
Yeah, it's exactly it, loss of the placenta. Where did it came from? The placenta. What do you think happens? Yeah, we know the baby cuts out. What do you think happens to the placenta? Yeah, it's got to come out too. Okay, trust me, it's not going to stay behind. Okay, so that's causing that gigantic drop. And then the placenta is out. And then what are we going to do with the placenta? I love it. Now, just like the previous group, I've actually prepared the last couple of years. Yes, we eat it, right? There's something called, have you guys had lunch yet? No. Ah, excellent. This is perfect. Okay. Placentophagy, eating your placenta. I swear to God, aliens that are out there watching the human race, they're laughing their asses off. Okay? So this is in fact what your placenta looks like. Okay? So do you really think when we talk about placentophagy or eating your placenta that we're sitting in the delivery room around a table, you get this served on a dish with a fork and a knife and you start chewing that down? That's not happening. Okay? The United States, as you all know very well, is a very capitalistic country and there's a land of opportunity. So there are companies out there that you can send your placenta to. And they will steam, heat it, dry it, pulverize it, and put it in a nice friendly capsule for you so you can actually swallow it. Okay? Why would we be eating our placenta? What is the point of eating your placenta? Nutrients. What do we think? That is absolute bullshit. Okay? Seriously. If I just completely annihilated the placenta to get it all pulverized, there is no nutritional value left whatsoever. Okay? Some people thought that maybe because of the iron content. It's highly vascularized, so blood, okay, that's fine. You're not getting more iron from your placenta than just eating healthy vegetables and some meat. Okay? But people swear by it. Okay? They feel really better and they feel good by eating some of these placental pills. What do we call that? Let's not call it crazy. What do we call that? Placebo effect. There's something called the placebo effect. I could have convinced the same people to take a water pill and tell it it's your placenta and they would have had the same sensation. There's absolutely no scientific evidence, zero, zilch, that this actually has any nutritional benefit or health benefits. Okay? Okay, sorry, I had to do this. I can't, this is just stupid. And then you get people talking about, oh no, we need to do this because animals do it as well. You know what cats and dogs do when they have an upset stomach? They eat grass so they throw up. So when you have an upset stomach, you're going to eat grass as well to throw up? Ah, seriously. Anyway, there's one more thing that I want to cover before tomorrow's lecture, and that is quickly lactation. Okay, so after birth lactation. This slide talks a little bit about estrogen and progesterone, how it helps with breast development during both, okay? We kind of already talked about this, but this slide is more important, okay? So, when we talk about lactation, there are two things. One, milk production, that is making milk. The second is milk ejection, which is getting the milk ejecting, or synthesis, okay, secretion, okay? The hormone that is important for milk production is prolactin. Okay? Prolactin comes from the anterior pituitary gland. Milk ejection is again oxytocin. Okay? So normally during pregnancy it's actually estrogen and progesterone that prevent release of these hormones. So you don't have milk production or secretion. Okay? And then obviously when progesterone levels and estrogen levels drop, prolactin is released, you make more milk, you have also oxytocin. And the stimulus for this are a variety that all act on the higher centers of the brain. That could be mechanoreceptors when the baby is suckling, but it could even be something as simple as the baby crying, okay? That act on the brain and you get these neuronal inputs, okay? Let's see. I did good, 2 minutes early. I'm all done for today. Okay, so tomorrow we have an invited speaker. It's actually another faculty member in our department, Dr. Winger. He actually used to work in an IVF clinic, so he's going to talk about IVF. So any question you have related to IVF, ask him. Okay, there's some real interesting ethical stuff. I'm sure he'll tell you some great stories about that stuff. Just kind of understand what IVF is. So I won't see you again. Have a great rest of your semester. Good luck next week, and maybe I'll see you around. Yes, thanks guys.
Okay, if it's stuck, why?